Anyway, I'm not talking about cocoa now, although at the session afterwards, that'll be there, and you can even taste some chocolate. What I'll be talking about today is biochar, and soon you'll understand what that is, in case you don't know what it is at the moment. Since we're celebrating 100 years to Machon Volcani Institute, I thought that it was appropriate to start this talk with a bit of a history lesson, so that we don't feel so proud of 100 years. Actually, we're going to go back a lot further than that, to Francisco d'Orellana, who was one of the first explorers of the New World from the Old World, and he was the first explorer of the Amazon River. And when he got back to Spain after his first journey there, back in 1542, well, he came back and told these amazing stories about these fantastic cities and streets paved with gold and made of white stone. And actually, the stories of El Dorado came from this era. And it seemed to people afterwards, which you will soon see, that he basically came back with these stories in order to get more money from the Queen to go back again and continue his explorations. He unfortunately didn't make it back a second time because he got into a fight in a pub in Trinidad and he was killed. So it took about 100 years before the explorers got back to the Amazon basin, and by the time they got there 100 years later, there was no evidence whatsoever of all the great things that he reported, nothing at all. And not only that, but it seemed like it was completely impossible that could happen because the soils looked like this. They were these incredibly infertile and barren ferrosols, and they could hardly sustain the hunters and gatherers there, let alone big civilizations the way he talked about. And he actually exits our story at this moment. He went down in history as one of the biggest liars of all time. And it was quite a long time went by before something started to come back, and still it seemed that perhaps there was something in what he said, and that was with the discovery of what's called Terrapretta, the Indio soils, which were discovered by a Cornellian professor, Charles Hart, who was actually an anthropologist, and he went on a journey in Brazil, and you can see that he wrote a nice book about it, and he discovered these pockets of very black, very rich soils, and also associated with them a lot of evidence of civilizations. You can see some of the things that he came back with. If you get to Cornell, you can go to the library. This is early version of PowerPoint presentations. You can see these huge murals on the walls in the library, in the museum there. And he saw these black, very rich soils, and he called these kitchen middens, which was basically, he thought that these soils were rich because people had been living there for thousands and thousands of years, and this was all the detritus of all their food in their kitchens and their refuse for a very long period of time. And it's, although as time went on, it became clear that, in fact, this Terrapretta is an artificial anthracite. It's highly fertile, very rich in minerals, and also, surprisingly, very rich in charcoal, which is the source of this black soil's color. It has lots of pottery, shards, bones, residues of manure, but they're quite old soils. They're anywhere between 500 to 8,000 years old. And to this day, they still maintain their fertility, although they were abandoned after a very short amount of time. It wasn't the 1,000 years that he had envisioned, but after 40, 50 years, these tribes would move on. And nevertheless, these soils are still fertile to this day. There were some early descriptions of Terrapretta. His student, Herbert Smith, followed him, and he talked about a field of cane that was growing there. You can see here in red, the cane field is a splendid sight. Stalks 10 feet high in many places and as big as one's wrist. This is the rich Terrapretta soil, the best soil on the Amazons. And after this also, there were a lot of farmers who went to South America from North America after the time of the Civil War, when southern plantation owners were not very satisfied with the result, so they decided to go to South America, and you also get reports from them. For example, this James Orton, the soil is black and very fertile. It beats South Carolina. That's where they grew rice. He says it yields, even without cultivation, 30 bushels of rice per acre. And these are these very rich soils. Turns out that such anthrosols are not limited only to the Amazon basin, but they found these types of soils all around the world, in South America, in Western Africa, in Asia. Here's a quote from an encyclopedia of agriculture in Japan from 1697. So you char your waste. You put also concentrated excretions, mix with it, manures, stock it for a while, and when you put it on the field, it's efficient for yielding any crop. So there was actually quite a wide use of putting charcoal into agricultural soils for improving the yield. This is a compendium of American agriculture from the 19th century. It was written in 1847. Charcoal dust was drilled in with seed. Has been found to increase the early growth from 4 to 10-fold. And actually, even not only in the US, but also in Europe, here they're talking about the use of charcoal for the disease, rust in wheat. Now, it came out right after the wheat talk, so there you go. So this was actually used in Europe as a way to repress this disease in France. And it was used into the 20th century also. Here they are selling charcoal for golfing, and also on the right in the timely turf topics, using forking and charcoal treatment. It's great for maintaining putting surfaces. You get a deeper root system, increased water efficiency, and so on. So it's very useful for golf courses. You can see early reports in the scientific literature comparing with growth without charcoal and with charcoal. And so this was really a very thriving and important topic. And then we had World War II. And in World War II, besides everything else we know about World War II, this was the advent of the chemical industry, actually. For example, DDT was discovered in 1939. So this was exactly at this time the chemical industry took off, also for global pesticide production and also for global fertilizer use. And so you can see what happened at that point. And at this moment, essentially, the use of charcoal for improving yields and for helping to protect plants against diseases was all forgotten. And then we'll just go ahead and fast forward to climate change. Starting in the 90s, a scientist who had done his PhD research on the Amazon soils and on the black soils and the Terrapretta soils in the Amazon remembered what he had done, and he started thinking about this whole problem of collection of CO2 in the atmosphere, and he started to promote the idea of putting charcoal into soils for helping to improve soil fertility, and also as a way to actually bury CO2 that starts in the atmosphere and is taken up by plants during photosynthesis, and then to manufacture charcoal from this material and put it into the soil. And so that idea started, along with other ideas of how to try and prevent or to mitigate climate change, substituting different biofuels like biodiesel and bioethanol and so on. So that was the way that charcoal is made is by pyrolysis. Usually the kind of charcoal that we're used to is charcoal on the week. Well, those of us who do a mangle on the weekend, then we have that on the weekend. It's a wood charcoal, okay? But the idea here was not to use wood to make energy, but it's actually to use this method called pyrolysis for waste biomass. Pyrolysis is a combustion under low or no oxygen conditions. So you don't get full burning to CO2. You get some CO2, you get some gases out, but you also get this solid material. And because it's not wood charcoal anymore, it got a new name, biochar. You can make it from all kinds of different organic wastes and waste biomasses. And on the one side, you can get out some hot gases, which you can use for energy and for chemicals and in industry. You get residual heat, which you could put it somewhere near, say, some buildings that you want also to heat or to use that residual heat for something. And then you get the solid fraction, which we now don't call charcoal, but we call biochar. Depends on the feedstock that you put in, but it'll be about 30% of what you initially put in. You put it back into the soil, where you then get improved soil productivity. And the half-life of biochar in soils is hundreds to thousands of years. So for our purposes, it's essentially a permanent carbon sequestration. So that was the idea that started to be promoted. The idea is this is even better than the other kinds of bioenergy fuels because it's actually carbon negative. You take something out and you essentially sequester it forever, and you get improved. So that was the idea behind the pyrolysis biochar platform. You got a lot for your money. Firstly, helping to mitigate climate change. You get to convert a waste into a product. You can generate energy, renewable energy, and also improve soil fertility. So when you hear people wax eloquently about biochar, it comes from this concept. I'm not going to go into how much of this is accurate, not accurate, what are all the okay, but what about here? Of course, it's not nearly as simple and as easy as this, but there is this whole concept to it. And what we did here is we looked into the whole part of improving soil fertility. And also waste to product, but I'll talk here mainly about the soil fertility. This is work that I did together intensively with colleagues from phytopathology, Yigal Elad, Norma Frankel, and a lot of other colleagues here. And one of the things that we discovered was that when you put biochar into the plant growing medium, it helps the plants to resist diseases better. And this was really quite unexpected. It was the first time anybody had discussed or found this sort of thing. The very first, I'm kind of afraid to press anything. It doesn't work anyway. Okay. Yeah, you think? Okay, right. Okay. I don't know. I didn't want to, do anything to the presentation. So you can see here some of the very first experiments that we did, the disease severity of a tomato to gray mold that was grown under ordinary conditions versus an addition of 5% in biochar. You can see similar results for powdery mildew with different types of biochars and made at different temperatures and from different starting materials. This was actually in a field trial that we carried out in Alava. Powdery mildew also in strawberry. You can see a very strong effect. I just want to mention now that this is not the control systems here and our experimental systems were all optimally fertigated. Okay, so this is not a nutrient effect. In fact, we checked and verified that in terms of nutrient levels and in terms of water holding capacity and so on, there's no difference whatsoever between the biochar growing plants and the plants that are growing without biochar. This is some extra additional effect, not nutrient and not water related. And then we continued, after looking at various foliar pathogens, to soil-borne pathogens. This is some of the work from Omer Frenkel. And we saw that, for example, that you got the same type of effect. You can see the differences as we change concentrations of biochar in the growing medium, and also even its effect on the extent of the Panama disease in banana. And so this was not only an effect that worked on foliar pathogens, but also on soil-borne pathogens. And you can see that there's also an effect of concentration. At some point, it can get to be too high a concentration, and it's not so. So it doesn't work all the time, every concentration, and not all the time, every, and each feedstock is going to be a little bit different. We were really surprised to see this. We don't work with pests, but they come to us, as we all know. And so this was just so happened that we got the broad mite, and Yigal went and he counted what was going on, and got this type of results. You can see that a little bit of biochar in the growing medium also seemed to repel the broad mite, and we saw this same result in the field trial in the Mont Parava. There were even various biochars that there was no infestation at all by the mite. 2 minutes. Okay, so plants also grow better. They're not just protected from disease, but they also grow better, as you can see in these pictures. Pictures are worth 1,000 words. And you can also see from the field trial that we did in the Arava, in the R&D station, in red, no biochar. And this is the best farmers in the world. We really didn't. We were quite surprised by these results, but this is the results that returned season after season, that even the best farmers in the world, by adding some biochar, we still got better, higher yields. And lastly, we went and looked at abiotic stresses, and this was actually work that we did together with Chaya Friedman, and looking at the effect of a biochar on acute heat stress. And I think you can see very clearly minus biochar plus biochar without the heat stress. Firstly, you get the same effect here. And then without biochar and with biochar with heat stress, you can see that there was a contribution of the biochar to helping to protect the Rhabdopsis against the acute heat stress. And we actually, what we see is this type of biochar elicited early acclimation state. And how we interpreted this, and it was based on quite a lot of intensive work and a lot of things that I'm not showing here. But we saw that essentially we got an early acclimated state where even without a stress, without what we called it was that biochar helps protect the plant against life's little stresses. I mean, we all suffer life's little stresses. We think we are giving the plant its optimal conditions, but do we really know? Okay, and so actually somehow the biochar elicits this early acclimated state, and then this it also takes the plant into protecting the plant in the acute stress state. So we have the biochar effect. We have plant growth promotion, we have systemic induced resistance to disease, and we have early acclimation to abiotic stresses. So don't worry. I mean, like, that's obviously the question, right? How do we explain the biochar effect? I'm like that lady there saying, this is really, really complex. How do we actually explain this biochar effect? But I knew that I'd be out of time, and so I'll just invite you all to come back in 100 years for our next centennial, and there will be the great reveal, and everything will be clear. So I just really want to say, I really want to thank my colleagues. It was this huge interdisciplinary group. It could not have done it in any other way. It's a great honor to me to be able to talk here at Israel at the Volcani's 100-year celebrations, and it's a great place, and I thank you also for your attention.
Thank you, Ellen. I'll take your invitation for 100 years.